students, this is Lynn, and today we're going to go into the synapses. So let's go ahead and get started. The synapse is a specific location where a neuron is functionally connected either to another neuron or to an effector. The synaptic cleft is this gap between the two cells that is filled with this interstitial fluid. You can see that in this case the neurons are very, very close, but they're not actually physically linked together. The dendrites receive information toward the cell body. The axon hillock is the trigger region where action potentials are then cascaded and fired. It travels along the membrane, and with the help of the myelination, it can skip via saltatory conduction and gets all the way down to the terminals to release the neurotransmitter. There are two types of synapses. A chemical synapse is one that is composed of a presynaptic neuron in which it is the producer or the releaser of a chemical, and the postsynaptic neuron is the one that receives the signal. These are found in the human body, and they're very common. This includes the synaptic knob, which requires this vesicle transport to dock onto the terminal membrane with the help of a series of proteins called the snare proteins. That will then allow the release of the neurotransmitter across that cleft, and it can then target onto a specific receptor. There is synaptic delay, which is the time that it takes for this neurotransmitter to bind to the receptor. On the other side, we have an electrical synapse. These are a presynaptic neuron that is connected via gap junction proteins to a postsynaptic neuron. These gap junction proteins are six-member proteins that link together like a staple. They're physically bound together so that when an ion flows, there really is no delay at all. These kind of synapses cannot be modulated, and the benefit is that it is much faster than a chemical synapse. Fast responses inside these receptors require a ligand-gated ion channel, so that means when the neurotransmitter or chemical binds to that receptor, the ligand-gated channel will open up, allowing an influx of ion or an efflux of an ion. So these ones occur really quickly. A slow response receptor involves a G protein coupled receptor. G protein is what we call the heterotrimeric 3 subunit protein that is on the intracellular side. It gets activated with the help of a guanine triphosphate nucleotide, and when the GTP is activated on the G protein, it will allow it to turn on an ion channel or an effector enzyme downstream next to it. These ones take a lot longer to activate than the ion channel themselves. Communication through action potentials. Pumps are proteins that move substances against a concentration gradient, which requires an input of energy. Our neurons are filled with these kind of pumps, the sodium-potassium pump and a calcium pump. On the other side, channels are ones that do not require ATP, and they are able to move down the gradient from the area that is high to low or from the inside out. They're oftentimes specific to one dedicated kind of ion. Leak channels are channels that tend to be open all the time. They're dominated by potassium channels that are found in the neuron. We also have chemically gated channels called CGCs. These are the receptors that respond to a neurotransmitter binding to that channel. And we also have voltage-gated channels abbreviated as VGCs, and these ones can respond to a change in the membrane potential. The VGCs have three states. They have a resting state where the receptor is not doing anything. We also have an activated state, which is meaning that it is bound to the chemical. And we also have one when the chemical is turned off and then called inactivation. So three states of these kind of VGC channels. In the resting state, that means the inactivation gate is open and the activation gate is closed. Therefore, sodiums or any ion cannot go in. In the activated state, that means the gates are open, and that means the ion can go right through the membrane into the inner compartment of the cell. When VGC is inactivated, that means the gate is open, but the inactivation gate is temporarily closed, so at that point, at the same time, no sodium can influx or exit. The distribution of pumps and channels throughout the neuron. So some pumps and channels are located throughout the entire plasma membrane of a neuron, whereas some others are predominantly located in a segment of the neuron's plasma membrane. On the right-hand side, you can see there's a cell body, and then we have the receptive segment, which is the processes of dendrites. We have the initial segment, which is towards the axon hillock, and then we have the axon tail, which is the conductive segment. Very bottom, we also have the transmissive segment that's not shown here, but that will be located at the axon butins and where the neurotransmitters are released. Sodium channels and potassium channels and NAK pumps are located throughout the entire plasma membrane. So in the receptive segment of a neuron, cell body and dendrites, you're going to find mostly chemically gated cation channels and some chloride ion channels. There's really no significant number of VGCs. The initial segment is conventionally considered to be the most important part, which is the axon hillock, that then allows the signaling to cascade onto the next neuron. This region is filled with both voltage-gated sodium channels and voltage-gated potassium channels. The conductive segment of the axon includes its axon and its branches, and it contains both voltage-gated sodium channels and voltage-gated potassium channels, just like the initial segment. However, at the transmissive segment, towards the synaptic knobs, you're going to find voltage-gated calcium channels and calcium pumps, and this is directly related to the releasing of the vesicle to allow the neurotransmitters to be secreted. Excitability and concentration gradients. So characteristics of a resting neuron. There are physics that involves with how the neuron's charge is distributed. The voltage represents the measure of a difference in electrical charge, and it is expressed in units called volts or millivolts or MV. There's going to be a current, which states that the current is related to the voltage over resistance. In other words, in physics, you have I is equal to V over R, and this is your classic Ohm's law, which states that the current is directly related to the voltage, but is inversely related to the resistance. In the neurons, these charged particles are ions, and the difference of the charge distribution is what we call the membrane potential. And as a result of a difference between the outside and inside, we have what we call the diffusion potential. So let's take a look at one ion first. In the bottom right-hand side picture in letter A, you can see that if the potassium ion were to diffuse through the membrane, the potassium ion is going to go from the inside of the cell, which is higher in concentration gradient, to the outside of the cell. That will then trigger a voltage difference of 94 millivolts with a negativity inside, so you express as negative 94. If you just isolate and look at the sodium ions by itself, sodiums tend to diffuse from the outside of the cell to the inside, creating a more positive side on the inside, making more charge on the inside, which creates a positive 61 millivolts. These ions are unevenly distributed across the membrane, and you have all these pumps that are actively pumping and moving ions around. So as a snapshot, the concentration of the cytosol, or on the inside of the cell, you're going to have a higher amount of potassium, and then you're going to have a much higher amount of sodium, chloride, and calcium on the outside of the cell. Over here on the upper right, you can see a chart showing you the concentration of ions distributed. The three most important ions that regulate the charge of a neuron are potassium, sodium, and chloride. Calcium is only relevant for the releasing of a neurotransmitter. So you can look at the numbers. On the outside of the cell, we have 5 millimolars of potassium, 140 millimolars of potassium on the inside. However, sodium is 150 millimolars on the outside, whereas only 15 millimolars on the inside. And chloride is 120 millimolars on the outside and 10 millimolars on the inside. So an experiment can be done by analyzing the charge difference between the outside and inside of the cell. You can take this microelectrode, place it inside a cell, and place the other end on the outside of the cell, and this voltage meter can measure the difference across the membrane. The resultant value is negative in that the inside of the cell is relatively more negative. There are two equations that we can use to apply to generate this kind of theory. The first is called the Nernst equation, which provides an insight into how a particular ion diffuses based on the concentration. The EMF stands for electromotive force of a particular single ion. It's equal to 61.5 MV divided by Z, which is the charge of the ion, and then multiplied by log 10 of the ion on the outside divided by the ion on the inside. So you can do this and plug and chug the ion concentration and also the Z charge for potassium, and then you generate a rough estimate value of negative 90 millivolts. You can then take that and plug and chug for sodium, you generate roughly positive 61 millivolts. And you can then apply that to get the chloride's value, which is roughly about negative 66 millivolts. The greater the ratio, the greater the tendency for the ion to diffuse in one particular direction. So that means the greater the Nernst potential, or the EMF, is required to prevent any additional net diffusion. So when we use this formula to analyze a single ion's EMF, it is assumed that the extracellular side on the outside has a zero potential. And so the Nernst equation is the potential on the inside of the membrane. So just be aware that the negative and positive values indicate from the perspective of the inside of the cell. There's another equation that could be applied to this concept called the Goldman-Hodgkin-Katz equation, which is taking into consideration of several different ions altogether. So you can also generate the electromotive force inside the millivolts expression. You have negative 61 times log of all the three ions that we just talked about in their concentration, either inside versus outside the cell. And based on this equation, you can see that there are three ions that are responsible for generating the resting membrane potential of a neuron. It's the sodium, potassium, and chloride. As a result of this equation, there are three characteristics that can be drawn. First of all is that the polarity of the electrical charge plays a role. It also depends on the permeability of what the ion is. If one ion is more permeable, then that ion determines the EMF. And then, of course, the concentration ratios respective of the inside and outside. So these ratios are fairly consistent, and for simplicity's sakes, we made it into whole numbers here. So the outside is 5 for potassium, and the inside is 140, and then the outside for sodium is 150, inside is 15. So you just take the outside divided by inside, then you get their ratio. So establishing and maintaining the resting membrane potential is really dependent on the distribution of those three ions, sodium, potassium, and chloride, as well as other substances, maybe phosphate ions and some components of organic molecules that are negatively charged, but those are very minor. The RMP is chiefly a consequence of the movement of ions across the membrane, but we also have to take into consideration that there are plenty of leak channels, leak potassium and leak sodium channels. The leakage channel for potassium through the nerve cell is probably the most important part. These channels are also called the tandem pore domain, also known as potassium channels, better known as leaky potassium channels, and they have a higher affinity to allow potassium to go through than sodium, even though sodiums can go through the same channel. So some sodiums are going to enter through the leak channels down its concentration gradient, and some potassium will diffuse out, making the negatively charged structures more on the inside of the cell. This movement is with the chemical gradient, but opposes that electrical gradient. Another factor that comes in play is the NAK pump, which is continually actively pumping these ions against each other. So on the right-hand side, you can see this is a visual of the NAK pump. It's a protein pump that utilizes ATP, and it moves 3 sodiums out for every 2 potassiums into the cell. So it creates this unbalanced number of ratios in charge. 3 positive charges going out for every 2 potassiums coming in. On the right-hand side, this is a visual of a potassium leak channel. And as you can see, the potassium has easier access to diffuse out, where sodiums could also go through, but it is less likely. The NAK pump has a relatively small role in maintaining the RMP, but it is constantly pumping, and as a result, for every single 3 sodiums are pumped out, only 2 potassiums are pumped in. And this accounts for about -3 to -4 millivolts difference to the resting membrane potential. The NAK pump does play a very significant role in maintaining this chemical conjugate gradient of NAK so that there's a lot more sodium on the outside than there are potassiums. The membrane potential in a resting excitable cell, like a neuron, is called the resting membrane potential, RMP. Three factors govern the RMP. If only potassium ions were to be in charge of generating RMP, you will see that the ratio of potassiums to the inside to the outside is 35 to 1. With the Nernst equation plugged in, you will generate that negative 94 millivolts. On the far right, you can see, imagine only potassium channels were utilized. That means there's 4 MEq per liter of potassium on the outside and 140. As a result of that, you would have negative 94 millivolts of charge. If only sodium ions play the role, and you take that consideration of ratio, it's 142 over 14, that ratio is 0.1. And when you plug in the Nernst equation for the inside of the membrane, you would generate 61 millivolts. You combine the potassium and the sodium, you will generate negative 86 millivolts. However, we also need to consider the NAK pump's contribution, which is about negative 3 to negative 4 millivolts. And so when you consider the movement of that 3 sodiums to the outside, for every 2 potassiums coming in, the net RMP is about negative 90 millivolts. The conductive segment of a neuron. The main activity of the conductive segment of the neuron, or the axon, is to generate an action potential. An action potential involves depolarization and repolarization, which is better termed as a nerve signal or a nerve impulse. A nerve signal or nerve impulse. The propagation of depolarization involves a sequential opening of these VGCs for sodium along the length of that axon. And this will then allow sodiums to flush right into the cell, meeting the threshold and then spiking up to a positive voltage. Then the polarization is the returning of the negative potential as potassium ions begin to exit through the voltage-gated channels of potassium. On the right-hand side, you can see that it looks like this. You start off at the resting membrane potential of about negative 90 to negative 70. The moment you trigger to the negative 70 range, it's going to start to allow sodium channels to activate. It goes up. That dotted line that you see of negative 55 represents the threshold. And the threshold is an all or none phenomenon that states that if the threshold is reached, an action potential will propagate. This will then climb up to a positive value of positive 30 of depolarization, and eventually recharge and then close the ion channel of sodium, and now potassium channels begin to open up and then recharging the neuron. It goes through repolarization and then hyperpolarizes down further before it resets. Over here is the visual showing you that as you start with the stimulus, you can see that the sodiums are not doing too much. During the red phase, after the stimulus is applied, the sodiums are able to rush in, and there's a rising in voltage shown here in green. And eventually it recharges where the sodiums are now closed off, but potassiums begin to efflux out. And on the right-hand side, you can see it's between the sodium channels and potassium channels doing their job. So here are the steps of the action potential. Number 1, a local current arrives at the axon hillock, which triggers voltage-gated channels to begin to open up. As sodium enters into the axon, causing it to reach the threshold, more numerous voltage-gated channels begin to open. Over here on the right-hand side, you can see that at the resting membrane potential, the channel is closed. The moment that you get close to the threshold, or when you are subthreshold, the door begins to open up and sodium ions are now able to enter in as it depolarizes. At the threshold, this is when the voltage-gated sodium channels are opened, and now depolarization occurs. Once you get to about 0 millivolts, this is when the channels are starting to inactivate and begin closing, and now the voltage peaks at approximately 30 to 35 millivolts. Step number 5 is when the voltage peaks, the potassium channels now are fully open. This will then lead to the rapid efflux of potassium out of the cell, reversing that charge so that this is going to make the inside of the cell more negative, and it's going down this graph. This is called repolarization. Step number 6 is when it goes all the way down below the resting membrane potential because these potassium channels stay open longer making it even more negative, and this is a brief period known as hyperpolarization, which is also what we refer to as the relative refractory period, in which another signal could fire, but it needs to overcome that slight difference between the resting member potential and that slightly more negative value. And then number 7 represents the reestablishment of the resting member potential as it recharges and awaits for another stimulus to hit. Over here is a visual showing you the propagation of the action potential. So this happens along the membrane of the axon. If this neuron had a myelination, then it could skip from one node to one node through a region called the node of Ranvier, and that will allow the signal to propagate much faster. But this is a moving charge, as you can see. As the sodiums rush in, it will then recharge, and then that action potential moves to the next block, and then down further of the axon segment, that will then allow sodium to rush in. On the right-hand side, you can see that when a neuron is myelinated with the fatter lipid insulation, it will, number 1, have a faster conduction speed. The thickness of a neuron fiber has a direct correlation to the speed that it can fire. So let's take a look at the speed and the size of the neuron. Let's say, for example, a 2 micrometer diameter neuron, you would have about 3 meters per second of speed. However, for a myelinated one of the same diameter, you would have close to 7. It's able to send a signal much faster. The refractory period is a brief time period after the action potential has been initiated during the axon, and it is either incapable of generating another signal or requires a much greater amount of stimulation to fire. The absolute refractory period is about 1 millisecond. This is a brief time where no stimulus can initiate because all the sodium channels are already open. At that moment in time, there's no other signal that could be fired. This ensures that the signal is going in one direction and moving towards the synaptic knob. At the peak height, about positive 30 millivolts, that's going to be your absolute refractory period. And then the relative refractory period is about 15 milliseconds. This drags on from the baseline of the hyperpolarization all the way to the next reset and the next threshold that a signal can fire. Some potassium channels are still open, the cell is slightly hyperpolarized, and at this point, you just have to overcome a greater amount of negative charge to be able to reach the threshold. During this phase, the voltage-gated sodium channels are usually in the resting state. So the VGCs for sodium are in the resting state. Graded potentials in a postsynaptic neuron are called postsynaptic potentials. These are short-lived changes in the RMP that has the following characteristics. They are established in the receptive segment by opening of chemically gated ion channels, and they can vary in the degree of change of the direction of the RMP. Graded potentials are expressed as 2 types. They can result in further depolarization. In that way, we call those excitatory postsynaptic potentials, also known as EPSPs. And if they produce a hyperpolarization, we call that an inhibitory postsynaptic potential, otherwise known as IPSPs. Numerous postsynaptic potentials can be generated because the postsynaptic neuron can bind to many neurotransmitters simultaneously. So you have the opportunity for neurons to convert together to one neuron or even divert out to multiple neurons. The neurotransmitter release from the presynaptic neuron can bind to a specific kind of chemically gated channel that can trigger it to open or temporarily allow the passage of a specific ion. And the degree of change really depends on the magnitude of the stimulus and the amount of neurotransmitter being bound per unit of time. Over here is a visual showing that EPSPs are depolarizations caused by entry of sodium, and you can see right here that it favors a depolarization and an action potential, goes up and comes down, whereas IPSPs favor hyperpolarization, which involves the exiting of potassium or the entry of chloride, making the inside of the cell more negative, dropping the voltage down even further. Over here is a visual showing you that one particular axon terminal can release a signal to that soma in the dendrite, and as the signal propagates, depending on the number of neurotransmitters bound, the signal can weaken, fade in a local aspect. So by the time it travels from the point of binding all the way down to the other end of the cell body, then it's going to decay. Summation of EPSPs and IPSPs occur at the initial segment or the axon hillock. This occurs by adding together all the postsynaptic potentials and determines if the threshold membrane potential is at -55 and whether or not the axon hillock will then commits to firing an action potential. There are two kinds of summation that can occur across space and time. The first kind is called spatial summation. This occurs when multiple presynaptic neurons release neurotransmitters at various locations on the receptive field, like we suggested earlier. If sufficient numbers of EPSPs are generated, then the action potential is going to fire. On the other side, a temporal summation occurs when one single presynaptic neuron releases a chemical that is excitatory at the same location repeatedly over and over to generate an action potential. Over here's a picture showing you that. You can see the threshold line, dotted line right here at -50. And the first orange tan color, the two depolarization stimuluses are spread out, so they're not going to add together. And temporal summation is when you have one single presynaptic neuron releasing chemicals at the same location repeatedly. So you have this additive effect. They're really close to each other, so now it reaches a threshold, and then you have a signal to fire. Over here in spatial, this is where multiple presynaptic neurons are releasing neurotransmitters at different locations, and then you can see that they do come together and eventually generate a signal enough to fire an action potential. You can also have an event where the EPSPs are released, and then you have an IPSP, which will even out and cancel out the signal. Over here's a chart for you to study to compare the graded potentials versus action potentials. A couple of key highlights here is that graded potentials can be summed, action potentials cannot be summed. The graded potential does not have a true threshold. The action potential does have a threshold, and then the graded potential does not have a refractory period. However, the action potential does. And on the bottom here, you can see the graded potential has either a depolarization or hyperpolarization, whereas an action potential can only depolarize if it is initiated by enough of a graded potential. All right, folks, thanks.